with us today. Julia received me with utmost warmth when I visited her at the Imperial College in London, just before COVID. Julia is a professor of cellular biophysics in the cardiovascular division in the UK Heart and Lung Institute at Imperial College in London. She's actually located in Hammersmith, which is one of the London suburbs, in a beautiful, really beautiful, functional, modern building that I really enjoyed seeing and visiting. Julia received her PhD from the Russian Academy of Science in St. Petersburg in cell biology. And she's now really working on cell biophysics of myocytes. And Julia has really done some fantastic work pushing the envelope on the resolution with which we study cardiac myocytes, going to subdomains in myocytes, in nanodomain resolution. So it's a nano resolution, looking at nanodomains and co-localizing the function of ion channels in the heart with nano resolution. So Julia has been really a pioneer in this area, pushing the envelope on the resolution and developing techniques for doing so. And she'll tell us about progress in her work on this in her seminar today. So Julia, thank you very much for joining us for the seminar.
Thank you.
Well, I ask everybody to mute themselves. That's number one. And number two, please hold your questions to the end and put them in the chat box, and I will read them at the conclusion of the talk. Okay, it's all yours, Julia.
Thank you very much. Thank you very much, Yoram, for your for your introduction. Yes, I'm trying to share screen, and it's very interesting because I practiced several times, and now 1 second, I will do it. It's very strange. Yes. Okay. Yes, it's here. Okay. So, could you hear me? So I think it's some of you already see some parts of my talks in the conference, in the symposiums, but I try to put all things together and show what we are doing now, and also show our just old work and new directions. So I will discuss about the functional nanodomains in cardiomyocytes. And first, I will introduce our technical advantages of our techniques for functional imaging. I'm working with the scanning and conductance microscopy, and this is really old picture when I try to explain the scanning and conductance microscopy. Is a filling the gap between the optical microscopy and electron microscopy. And using our technique, we can resolve the small features on the surface of the different type of cells with high resolution. And of course, now we use not only scanning and conductance microscopy, but also a lot of different combinations. So we use the glass micropipette as a sensitive probe, and we scanning just the live objects. We have our pipette on the piezo element, and we have 2 electrodes, one inside the pipette, another inside the solution, and we measure the current between two electrodes. And also important things, we keep the distance between the pipette and cell always constant. And during the scanning, we pick up the vertical displacement of our pipette together with the current. And we produce the topographical map of our cell. And this is a current image. This is a current image, and in this current image, we can just see some sort of feature of cardiomyocytes in this situation. Our resolution depends on the diameter of our pipette, and we can use the very sharp pipette, we can use not very sharp pipette, and potentially I will discuss today why we're using the different pipettes of different diameters. So when we start to work with this scanning and conductance microscopy, we start to work with the simple cells, with the kidney cells, and we check our image with the electron microscopy image, and we just really see this. With our imaging, we can resolve the small features, and also with our imaging, we can just measure the height and volume of the cells. Also, we just, in 2009, we update our system and we start to use the Hopin mode microscopy. When our pipette hopping from the fixed distance, and with this hopping mode, we can resolve not only the small features, we can just scan very thick objects. As a tissue, for example, now we can scan the open aorta, and potentially because our pipette just hop from the fixed distance. We name this as a Hopin mode scanning and conductance microscopy. So in this Hopin mode scanning and conductance microscopy, we can just resolve the small features. This is live cells in the collagen matrix with the high resolution. This is a small high resolution resolving of the collagen fibers, for example. So okay, sorry, I'm trying to move. This one is we can resolve the tissue with this Hopin mode pipette and the new program, and we can just, this is a stereocilia, and this is a microvillia from stereocilia, and this is a scanning electron microscopy image, and this is our imaging when we can just resolve the small features in very, very tiny in the surface of the stereocilia. Also, this we can check the surface of the cells after and during the stimulation, and we try to add the hypertonic medium solution and hypertonic medium solution to the live cells, and we can just see the beauty of our system when you can just follow to the same cells. You have cell number 1, cell number 2, and when you add the hypertonic medium stress there, you can just really see this how changes surface on this exactly these cells. So this is a live imaging. When you can just add and do the hypertonic medium stress, you see the how you can lose the integrity between the epithelial cells. So also this we can just scan long time, and we can just see how cells change the volume, and this is just also epithelial cells, how the cells can change the volume and shape during the 24 hours. We can see this with our system, how cells can divide, how cells also can just when you can add something, how the contact change, how the volume change. This is just really this is what happened during the 24 hours. Of course, this is just several seconds of this movie, but potentially you can see this how cells change behavior. So also very interesting, this we can just check how if you add to the monolayer, this is for example skin cells, when you add the calcium, you can see how you can establish the contact between the cells. But it's very, very important to try to see this how you can just monitor the alive cells, and after that you can just have all these images and change the changing in volume after calcium addition. Of course, it's important we can just resolve the small features on the surface of the cells, and we start from the epithelial cells, and we study the movement of the microvilli, and we just saw the formation of the microvilli, steady states, and retraction of the microvilli. It's known that on the top of the microvilli, for example, in the lymphocytes, you have a CD44 sitting on the microvilli, and a lot of channels and receptors sitting on the microvilli and the kidney cells. And in this time, I started to think about the compartmentation, about the not all channels and receptors just try to distribute randomly on the surface on the different type of cells. And from this time, I start to think about compartmentation and channels and receptors. So also we can just use the very tiny pipette and try to image the very small features such as the cluttering coated pits and the dynamic of this cluttering coated pits on the surface of the different type of cells. And also very important, we can resolve the caveoli-like structures, which is signalosomes, and have a caveolin, and this is signalosomes are very important because a lot of channels and receptors sitting inside this caveoli-like structures, and this is very important for cardiomyocytes. And from this time, so just I can just tell you about the advantages of just scanning. So we can scan surface of live cells with high resolution, study changes of the topography of the cell surface during cell life, and measure cell volume and height, and study the dynamics of cell surface structure. And I did scanning of different type of cells and employed a lot of different methods to measure the functional different behavior of different cells, from sperm cells, neurons, kidney cells, melanocytes, fibroblasts. But potentially for me, it was very, very important to study my favorite cells and cardiomyocytes, because the cardiomyocytes very good structured cells, especially the ventricular cardiomyocytes, and they have a tubule open area, the center of excitation, contraction, coupling, Z groove, and sarcomeric structures. So potentially when I start to work with cardiomyocytes, we just really compare the cardiomyocytes with the TM images, and we found that we can resolve this Z groove and tubule open area. And we start with comparison of cardiomyocytes, normal cardiomyocytes, and cardiomyocytes from diseased heart, from 16 weeks of coronary ligation, from cardiomyocytes from myocardial infarction rats. So we found these cells have a very low tubule index, and also we introduce new Z groove index. When you have a good organization of the cardiomyocytes and Z line, we just really have a high Z groove index. When you have a very low organization of Z groove, you have a very low Z groove ratio. And we found the cells after myocardial infarction have a very low Z groove ratio in tubules. And in parallel, we check they have differences in the in the calcium transient and spark amplitude and spark frequency. But in this time, we study the cells in one population, we study the structure of cells in one population, and we study the function of cells in another population. And from this point, I decided to combine the several techniques to study function and structure in one goal. So potentially we now study the tubule areas. We name the distance between the tubules as a crest area. Also, we can just resolve the intercalated discs of cardiomyocytes and the membrane invagination, as I already told you, the caveoli-like structures. So we are trying to answer the questions: how the signal nanodomains are organized and where are they located? What is the relationship between the structure and function in these compartments? And how these compartments change in heart failure? And do changes in structure of compartments in heart failure affect their function? So we just combine and design a lot of different techniques to answer this question, and we use our system for scanning and scanning with a high resolution. We also can scan in angle and resolve the intercalated disc, and also we can just measure something with this intercalated disc area and different tissue, because we introduce the angle approach scanning and conductance microscopy. We use the combination of scanning and conductance microscopy and confocal microscopy for checking some fluorescent features on the surface, and also do the 3D reconstruction of opening tubules and tubules inside to do the 3D organization of tubule structure in the different type of cells. We also introduce the combination of scanning and conductance microscopy with the FRET technique to study the movement of the different type of receptors, and the study the cyclic AMP signaling and cyclic GMP signaling. We also can use this pipette as a pressure jet and measure the stiffness of the cardiomyocytes, and also we can just combine the stiffness measurement with the calcium imaging and measure and study the mechanosensitive nanodomains. Also, we recently just introduced the mechano scanning and conductance microscopy when in one goal we can just scan the cells, cardiomyocytes, and during scanning we apply the pressure, and we can say this, we can just really produce the mechanical map of the cardiomyocytes together with the topographical map of the cardiomyocytes. Recently, we also try to combine the optical stimulation and contractile measurement, and measure and study the contact between the fibroblast cardiomyocytes, and employ the optogenetics when we can just, for example, infect with channel rhodopsin fibroblasts and activate them with light and measure using the pipette the contraction of the cardiomyocytes. Also, we start to work and use the nanotweezers technique when we can just really can scan and can use the pipette and take the probe and try to see the different, not only one cell, just RNA-seq, also try to check from our compartment, take the genetic material, and this is very cool, and we are just really will show very soon our results and our new achievements. But my favorite, and I think probably favorite in this audience, the super resolution scanning patch clamp, when we can scan the object and after that use our pipette and try to record the ion channels from the area of our interest. So I will discuss about the sum of this combination. I don't have time to discuss about all of them, and I will discuss about just mostly the scan and thread and super resolution scanning patch clamp, and potentially the SMILE study the localization of beta-adrenergic receptor signaling to surface structure of the cardiomyocytes. And I think it's, this work about a different distribution of beta-adrenergic receptor in the cardiomyocytes and changes of this distribution during the heart failure. But we are studying now a lot of things about the distribution of beta-adrenergic receptor. We study the difference of organization of beta-adrenergic receptor and different type of cells, and for example, the differences in the localization of beta receptors between the apex and the base of the heart, and also but direction, the localization of beta-adrenergic receptor is a really important direction of my lab. Another direction of my lab, the special localization of L-type calcium channels and modification in heart failure, and the coupling of L-type calcium channels to the beta-adrenergic receptor and is it change in failing cells. So first of all, I will discuss about beta-adrenergic stimulation and also mostly about cyclic MP. Just distribution and diffusion, and also today I will discuss about our new work about localization of beta-3 receptor and about cyclic GMP in heart failure. So how it was from very beginning, we knew the beta-1 and beta-2 receptors have a different function but have a similar second messenger cyclic MP, and we didn't know how we can localize this receptor, and we decided this do the combination of scanning and conductance microscopy and the thread technique using the just fluorescent sensor EPAC2 comp, which change the conformation when the cyclic MP bind the binding domain. And we just produce very simple experiment when we infect the cardiomyocytes with this virus, with this fluorescent thread sensor, and we just activate the beta-adrenergic receptor through the pipette and just measure the thread in the local direction. And potentially this we have the agonist of beta-1 or beta-2 receptor inside the pipette, and potentially this we first scan, after that we apply pressure or voltage. Now we applying only voltage because it's more local, and when we apply our agonist, we just really cyclic IP goes up and we have a drop in thread, and we found the beta-1 receptor just located everywhere in the crest or in the T-tubule, and beta-2 receptor just located only in the T-tubule, no beta-2 receptor in the crest. But in the heart failure cells, in our model of heart failure, we found the beta-1 receptor the same, and beta-2 receptor also go to the crest. So we found that if in healthy cells we have a very local signal of beta-2 receptor, in the failing cells we have a global signal of from beta-2 receptor, and we found the just PK, the buffer, the cyclic MP, and we found that in heart failure cells we have a redistribution of the R2 subunits of pKa, and we just suggest the functional beta-2 receptor normally located in the T-tubule and local signaling in the T-tubule confined by interaction with the R2 subunits of pKa, and in heart failure, beta-2 receptor redistribute from T-tubule to the crest, and redistribution change the mode of cyclic AMP compartmentation from localized to global signaling. And now this we are thinking about, can we reverse the beta-2 receptor redistribution in heart failure? And we just check the role of caveolin-3 and caveoli in this process. And we found when we just transfect the cells with the caveolin-3, additional caveolin-3, we have a more caveoli in the cardiomyocytes. And when we infect the cardiomyocytes with dominant negative caveolin-3, we have less caveoli in the cardiomyocytes. And we found when we have a caveolin-3 dominant negative, we don't have so local signal from the beta-2. And we decided to just infect the heart failure cells with the caveolin-3, and we found that after infection and after introduce the expression of caveolin-3, we go for the local signal after beta-2 stimulation. And we just conclude that caveolin-3 plays a crucial role for the localization of beta-2 adrenergic receptor and compartmentation of beta-2 cyclic AMP signaling to T-tubule of healthy cardiomyocytes, and overexpression of caveolin-3 in failing myocytes can partially restore the disrupted localization of this receptor. So we also was very interested to understand the role of beta-2 receptor dependent signaling localization during the myocardial infarction. When we have this detubulation, when we have this redistribution, and we decided to check the progressive changes during the heart failure and during the myocardial infarction. We checked the 4 weeks after a myocardial infarction, 8 weeks and 16 weeks, and we found that just we have a gradually change in the T-tubule changes and in the beta-adrenergic receptor signaling. So we found already in the 4 weeks after myocardial infarction, we have a lot of longitudinal axial tubules in the myocardial infarction cells. And also this, we have their severe detubulation already in the early stage. But also very interesting, we have a lot of caveolin-3 overexpression in the myocardial infarction, but this all caveolin-3 in the cytosol. The caveolin-3 during myocardial infarction is not functional in membrane. A majority go to the cytoplasm. And also interesting that we have a lot of signal of beta-2 receptor in the control, but potentially this we have some sort of changes already from the 4 to 16 weeks. And here this we have schematic how what changes we have during the myocardial infarction. Already in the 4 weeks, we have a lot of axial tubules and longitudinal elements in cardiomyocytes. But caveolin-3, for example, and beta-2 is still in the T-tubules, and caveolin-3 mostly in the membrane. But already in the 8 weeks, we have some caveolin in the cytoplasm and beta-2 receptors, some in the T-tubules, some in the crest. In 16 weeks, you already have the fully destruction, and you have the all caveolin-3 in the cytoplasm and beta-2 receptor everywhere. So potentially the surface structure and transverse tubule change begin earlier after induction of myocardial infarction and partly compensatory until cardiac decompensation sets in. And the functional beta-2 receptor delocalization develops slightly later, at the phase associated with the transition to decompensated heart failure. What interesting we found recently, we found the regional difference in beta-2 receptor signaling in the left ventricle. And everybody knows the beta-2 responsiveness in Takotsubo syndrome when we have a apical ballooning in the apical part of the heart. And we found some sort of discrepancy of our results, because we found that when we isolate the cells from apex, the beta-2 stimulation just increase compared with basal cells, but from another point of view, we didn't find any differences with cyclic AMP between apex and base. And we just really start to think why, because we decided to check the structural features of the cells from apex and base. And we found the cells from apical part very different from cell from basal part of the heart. We found if we have a not different the Z group index, we have a power of tubule regularity and density different, and also very important, apical cells have 5 times less caveoli than basal cells. So potentially in the heart we have cells with the different structures from apex and base. And we also, in order to see the beta-adrenergic cyclic AMP signaling, we decided to use the local Fred sensor. We use not only cytoplasmic sensor, we use the sensor plasma membrane sensor and sensor located to the R2 subunits of PKA. And we found that if we don't have any differences in the cytoplasmic sensor between the apex and base, when we remove, so when we just really use the R2 sensor, just local sensor for PKA activity, we have a differences between apex and base. And this difference, just when we destroy the caveoli, we don't see this difference anymore. After that, also important when we use the membrane sensor, when can just really measure the cyclic AMP only in the membrane, we have a dramatically drop of the just cyclic AMP between apex and base. And after removal of caveoli, we have the same equalized our signal. So it's very, very important, and we check also the main PDE of this process, PDE4, and we found we have a PDE4 more active in base part of the cells. And we just conclude the basal cells and apical cells are different. Basal cells have highly organized microdomains, in particular caveoli and tubules, and that allows to tight control of beta-2 mediated cyclic AMP production. And addition of PDE4 is more expressed and active. And apical cells have less caveoli and less organized tubule, which led to the beta-2 mediated cyclic AMP being able to reach contractile proteins and modulate contractility. Removal of caveoli and inhibition of PDE4 equilibrates the apical and basal beta-2 mediated cyclic AMP production, as well as contraction response to beta-2 stimulation. So this part of the work about the beta-2 mostly just localization and beta-2 function and cyclic AMP release. But now it's really very important to study the cyclic GMP, because this is already clinical trial and a lot of drugs was developed and just really stimulate the cyclic GMP in heart failure. And our last work, we found the beta-3 receptor redistribution in the heart failure and we found the complex which just really go beta-3 effect impair the NO, cyclic GMP, and PGE2 signaling in failing cardiomyocytes. So potentially this we study the beta-3 localization using the not cyclic AMP, of course, sensor, cyclic GMP sensor, and we would like to study the exact submembrane localization of functional beta-3 in adult cardiomyocytes and special temporary regulation of the cyclic GMP signal and their potential interaction with cyclic AMP signaling pathway, and also to study the alteration of beta-3 signaling in failing cells. So first of all, this we just use the cyclic GMP sensor with the two fluorophores Sapphire and Dimer 2, and with the binding domain which just can bind the cyclic G. And when we activate with ISA cardiomyocytes, we see the very high release of cyclic GMP, and this release the decrease in failing cardiomyocytes. Also very interesting, when we block beta-1 and beta-2 receptor, we don't have any changes. So this is really the signal from beta-3 receptor. And very important, when we add the antagonist to beta-3 receptor, we don't have any cyclic GMP release. And also when we block NOS, we use the L name for block NOS blocker, we don't have also any cyclic GMP release. So very important, we check the activity of the main PDE in this process, and we found the main PDE in this process PDE2 and PDE5, and this PDE2 increased during the heart failure. So after that, we tried to do the local application of beta-3 stimulation, and we found the beta-3 active in the T-tubules and not in the crest. They're just similar with the beta-2 receptors, but in the heart failure, we have a lot of activity in the crest. So we use in our work the special peptide, that C3CD peptide, which binding to covalent-3 specific scaffolding domain, and we disturb this caveoli signalosomes. And when we disturb the caveoli signalosomes, we found we don't have anymore the signal in the T-tubules and not signal in the crest. When we disturb the caveoli microdomain, we have the equalize the signal of cyclic GMP release between T-tubule and the crest. So also we check the signalosomes, and we just stain with soluble glutinous cyclase and covalent-3 and see the differences in this complex during heart failure. And very important, we check the crosstalk between the cyclic A and cyclic G when we use the cyclic AMP sensor, and when we just activate adenylyl cyclase with phoskalin, and we just after that activate the beta-3 receptor, and we see the decrease in cyclic AMP release, and this will abolish of blocker of PDE2. But in the sharp contrast, in the heart failure, you don't have any just depress of the cyclic AMP release with the agonist of beta-3 receptor. So potentially this is our schematic, and we just can conclude in healthy red cardiomyocytes, functional beta-3 receptor localized exclusively with the transferred T-tubule and stimulate cyclic GMP pool. In failing cells, beta-3 adrenergic receptor cyclic GMP signal decrease in the T-tubules, and cyclic GMP pool is predominantly regulated by phosphodiesterase 2 and 5. And in healthy cardiomyocytes, beta-3 stimulation decreases overall adenylyl cyclase dependent cyclic AMP level, but PDE2 mediated cyclic G, cyclic A crosstalk. And in heart failure, cyclic A crosstalk is disturbed, and beta-3 stimulation no longer has a significant effect on overall cyclic AMP levels. So This is what really I would like to say about cyclic G, but now I would like to show our work with the L-type calcium channels localization. So we developed the super resolution patch clamp long time ago when we used the small nanopipette, when we first scan the area of our interest, and after that we just use the small nanopipette with the high resistance and try to record the ion channels. But recently we developed this system, and we first used the sharp pipette. We just try to just do the topographical image with sharp pipette. After that we clip our pipette, but now our diameter of the pipette around 200 nanometers. We cover the tubules, but we can just really record the our channels from the area of our interest, from tubules or from the crest. So we try to record the channels from the tubules and from the crest, and we found the majority of functional L-type calcium channels located in the tubules, and not a lot of channels in the crest. In contrast, we check the atrial cardiomyocytes, and we found the atrial cardiomyocytes have a different groups with the good organized surface structure, with the disorganized surface structure, and no surface structure. And we found in atrial cardiomyocytes, in rat and human cardiomyocytes, we have L-type calcium channels in tubules, also in the crest, in the same proportion. And we found because atrial myocytes have 5 times more caveoli than ventricle myocytes, and when we add the cyclodextrin, which disturb the caveoli, we cannot find the channels in the crest. And also we use the special REM cuff, which was developed in Stephen Hauser laboratory, and when we just really add this, we prove that our channels have the channels located in the caveoli in atrial cardiomyocytes. And we just potentially found the LTCC distribute equally in tubule in the crest in atrial myocytes, and functional LTCC in the crest eliminated by caveoli targeted antagonist or disrupting caveoli with the cyclodextrin. And also we provide the first direct evidence for the existence of 2 distinct subpopulations of functional LTCC in rat and human atrial myocytes. Also, this what happened with ventricular myocytes. In ventricular myocytes, we also check the left and right ventricular cardiomyocytes, and we found that also not only apex and base cells are different, also left ventricle, right ventricle have a different organization of tubule and density of the tubule. And also this we have differences in Z groove ratio. Also this we found compare with left ventricle and right ventricle, we have a lot of channels in the crest, so potentially they behave as a just failing cells. So we found right ventricle myocytes have less dense and less regular tubule structure compared with left cardiomyocytes. Sarcolemma topography is less organized. And also important, LTCC distribute equally in tubules and crest areas of the sarcolemma in right ventricle myocytes, similar to atrial myocytes. What happened in this heart failure? So it's very interesting. We found when we use our model of myocardial infarction, 16 weeks of coronary ligation, we found the distribution of functional LTCC is different in failing cardiomyocytes. We found in just rat and human also, we found that we have in failing a lot of channels in the crest, and these channels have enormous high open probability only in the crest. And also very interesting, we found this high open probability channels you can close with the blocker of CAM kinase 2. And more interesting, we just give this results to our collaborators in Baltimore, Natalia Troyanova lab, and they just do the some mathematical modeling. This is just propagation in simulated control. They use our data of these channels in nanodomains, and they found in the control you have a normal propagation, and in the simulated failing you have some sort of re-entry. And it's very, very interesting and important, because now we start to work. So we conclude the active LTCC channels are not uniformly distributed along the surface of the cells, and disruption of membrane structure this led to the these channels which have a very high open probability. And very important, this high open probability channels can be linked to CAM kinase 2 phosphorylation of L-type calcium channels. And also translational modeling study suggests that this specific LTCC could be potential target of arrhythmia. So, of course, what's happened in the patients? So we now have access to the transplant program, and we use the cells and also slices from the patients with dilated cardiomyopathy, with ischemic heart disease, and also patients with the left ventricular assist device, and just which can pump blood in the aorta to the body. And we use the cells from all these patients, and also cells from patients with LVAD. And we found the potentially we have different populations during the patient's failure. So what we found the cells from the patients with ischemic heart disease, with dilated cardiomyopathy, have the very low density of the T-tubules and power of T-tubule regularity and Z-groove index. But after LVAD, sometimes you have the cells with increased the T-tubule density. What happened with the L-type calcium channels? This is very, very interesting data. We found in the different diseases, we have a different mechanism of our L-type calcium channel distribution with the high open probability. And ischemic heart disease, we have a very high open probability in the T-tubules. In dilated cardiomyopathy, we have this disease channels in the crest with a high open probability. And very important, LVAD can just close these channels and decrease the abnormal open probability. And what the mechanism of this? So potentially we check this when we have the higher open probability in ischemic heart disease. When we use the PKA blocker, we can close these channels. And dilated cardiomyopathy, the opposite, we can close the channels, abnormal open probability channel in the crest. We can close this channel with the blocker for CAM kinase 2. And potentially this also important, Natalia produced models and show the difference. So in ischemic, we have EAD, and in DCM, we have re-entry. And potentially this is our conclusion from this work, from the nanodomains to the whole heart. And we can just conclude the etiology of the human disease determines the special localization and function of LTCC, and unloading the heart through the LVAD implantation normalize the differences observed between the ischemic heart disease and DCM patients, which suggested that electrophysiological remodeling is a plastic process that can be partially controlled. And also important, translational modeling study suggests that controlling the phosphorylation of LTCC through PKA CAM kinase pathway in disease-specific way could lead to new treatments. So now this we are doing the work and study the coupling between the L-type calcium channels and beta-adrenergic receptors, try to activate the receptor through the pipette and as an output to record the channel's activity. So this is our future direction, to study the PD in this process and also check the LTCC recording and intercalated disc, and also study the activity in the human patients. And this is my old paper together with Mario Del Mar, because this we first time use our angle scan, and we found nanoscale visualization on functional nodes in intercalated disc. And in this study, we just really check the sodium channels and intercalated disc of cardiomyocytes, and we found the all sodium channels, just active sodium channels, just in the big clusters. And in other 70% of all patches, we don't have any channels in the patch. So also this we show this we can do the very high resolution angle images of intercalated disc and try to check them in the just high resolution different other techniques, and we just really have the same clustering. So potentially we can scan in the intercalated disc and also resolve the structure of intercalated disc and combine with the patch clamp to study channel distribution of the intercalated disc. So we can just use our technique with this several combinations of technique to study a lot of diseases in this cardiac area, from atherosclerosis to heart failure, arrhythmia, hypertrophy, mechanical dysfunction, and also valve disease. And I think it's important to think about nanodomains in cardiomyocytes and different cardiac cells, and compartmentation of signaling and compartmentation of beta-adrenergic receptor signaling and L-type calcium channels. And this is photos from my lab, from my old members, when we discussed how big nanodomains are. And this is just acknowledgement for my collaborators from Imperial College, especially Professor Shank Hardin, Yuri Korchev, and also my previous trainee, Professor Vyacheslav Nikolaev and Dr. Michele Miragali, and just Professor Natalia Troyanova as a main person who helping to do the modeling work with our results. Also Professor Stephen Hauser from Temple University, who we work together in this L-type calcium channels studies, and also Mario Del Mar from New York, from States, this intercalated disc study what I show you today. So thank you very much for your attention, and please ask any questions. I think I'm okay with time.
Okay. Thank you, Yulia, for a tour de force through tremendous amount of data. It'll take some time to absorb it all, because it really involved many different types of data, from fascinating, new and powerful and elegant tools. Before I look at the chat for other people's question, let me ask one question that is on my mind. And this is, you showed some differences in beta and beta function from apex and base, and also heterogenic distribution of L-type calcium channels. Did you look at the right ventricular outflow tract? And the reason I'm asking is because the RVOT is sort of a hotspot for arrhythmic activity and arrhythmia generation. It'd be interesting to see if there is something unique about the RVOT.
Not yet, because I thought about it, because it's when we start to do the RV, I think it's now time to do it, because it's honestly, we didn't expect these differences in heterogeneity inside the heart and even in the ventricular cells. That's why, yeah, it's a good idea. Thank you very much. I think it's really good things to check it, because it's, yeah, I think it's a different compartmentation. Yeah.
Because it also has different growth structure. You know, it's thinner,
Sure.
comes from a different field in evolution, in development.
Yes.
And so it'd be very interesting to see the differences. Okay, let me look at the chat. And the first question is from Jean-Pierre Benita from Paris. And Jean-Pierre is asking, did you check whether you have differences in localization of beta-2 between epicardiac and endocardiac cardiomyocytes?
No, because why we study apex base? Because we are just really interested in Takotsubo, and this is some sort of beta-2 interest and gradient of beta-2. And epicardium, no, we didn't. We just really don't have this aim and task, but potentially it's possible to check. I think it's also maybe have difference, because this is also, I know electrophysiologically this is a big differences between epicardial and endocardial. Yes.
Okay, next, Igor Efimov. Oops, it jumped. Igor Efimov is asking, first he's saying great work, and then he's saying, can your technology be used for long-term studies in myocytes? And what he means by long-term, he says multi-day.
Long-term for myocytes, what it means? So in culture,
yeah,
or
maybe I clarify. Yeah, if you study, for instance, you know, conditioning of myocytes by various stimulation protocols, pharmacological, etc., but doing it longitudinally over 1 week, 2 week in culture.
no, 1 week, 2 week, but during the culture.
In culture.
Yeah, but in culture, your myocytes, of course, you can do the stem cells or something, but your myocytes, okay, lost the structure after 72 hours, let's say, but we can scan, because we have now thermochamber, all this stuff, check the pH and CO2 or something. You can scan. We scan, for example, differentiated cells, stem cells. We scan the dividing cells. You can scan 3 days. Yes, you can. But cardiomyocytes, your structure after 3 days will change.
No, unless you culture it incorrectly, then of course it will change. But if you
but you lost during cultivation in ventricular myocytes, you lost the tubules after 72 days. So what? Slices, yes, you can scan slices, yes, fine. But isolated myocytes, of course, you will lose the structure after 72 days. I don't know what myocytes you are using if you not lost structure after 72 days.
No, I mean engineered heart tissues, for example.
Okay, you can scan. Yeah, you can scan several days. Yes, no problem.
Have you done it yet?
Yes, I did. No, I didn't do the cardiomyocytes. I did another different cells types. For example, for 72 hours, divided something. But for cardiomyocytes, not yet. I didn't have this, let's say, purpose for this, but I can do it.
Okay, thanks.
Okay, Guy Salama. When you compare.
Hello, Julia. It was a wonderful talk. I remember hearing you before, but as always, new stuff, and it's always very exciting. So my first question was a little bit of a localization issue in the heart failure studies. Where were the myocytes taken? Were you able to discriminate between border zone, MI zone?
Sure,
Are you
sure,
looking
sure.
far away?
Sure.
I wasn't
But
sure. And that's
because potentially in this, because now we are trying to divide this. This one is more or less, this is something just what cells survive. So this is not very close to border zone because it's a big differences. Now we're trying to, it will be very soon, we just will publish very soon the different organization from border zone distance and distance very close to border zone, or this one is unfortunately all together, but it's not very close to border zone, because now I know we have a big differences with cardiomyocytes nearby
Right.
border zone and just long distance
Can you
from border zone.
detect the structure of the SR? I'm asking because we're studying cell division in myocytes, and there are very interesting structural changes in terms of calcium handling. Is it possible to detect SR?
I don't
deep.
know, because
Yeah.
unfortunately we have the this is our big limitation, because this is a surface. We can take the
electrode
pipette
won't go
inside the
in, but
tubules, but potentially what we did the very interesting things. We did the prefixed, some sort of very long, very low fixator when you can just really open the membrane. But
right.
this is, yes, but for these particular things, functionality of your study, you know, even you have very low fixator, very low, you still can open, you can see something, but how functional it will be, I don't know, because honestly, I'm doing the functional microscopy, I'm just doing the live channels or something. I don't know if you can just change the sarcoplasmic reticulum something, and after that they pick up the channels or sparks or something. This is, you can really modulate something, and you can just really play with something in sarcoplasmic reticulum, and after that as an output we can just the same things what we're doing with Fred, because Fred is inside, but what we're doing, we localize the signal, and after that we measure inside. Also, this you can just localize your signal, do some sort of signal from outside to go to inside, and after that to measure the inside. Yes.
Right, that would be an idea, but perhaps the T-tubule positioning is changing in cell division. That's something that you might have already done. And I don't know if you have data on that we can
we are doing this, let's say, lability of the tubules. This is true.
Right.
This because honestly, our idea was the tubule so stable, the opening, all this stable. We're doing all this. Sometimes it's not really true, because potentially when we have the ideas about lability, and when we have the confocal and just confocal and scanning together, and you can just see the confocal, you can just switch off feedback and do the confocal things and see this how these tubules can be dynamic. Yes, we are trying to do it. And the second, what we are trying to do in this really long time, we can just inject through the pipette to the individual tubules the dye, and this is also tricky business, but we are still doing this. Yeah, we are trying. Yes.
Great work. Thank you.
Thank you.
Thanks, Guy. Let's go to the next question. The next question is basically the same and was covered. It's from Chinzu, and the question is the following. Thanks for the great presentation. For post-infarction hearts, are there differences between the border zone and remote zone in the cell structure and channel distribution? So,
yes. It's different, but potentially they did not finish to study this, and I think it's very soon we will show this. And yes, the cells are different, and a lot of, again, this viability of cells is very different. And yes, and cells, when cells survive, this is also another point, and we need to just really assess this population. I hope I will show this very soon. Yeah.
I meant to ask you some about the simulation. The simulations were the ones that were done by Tomo.
Natalia. With Natalia, yes, I don't know.
But that's the paper that came with Tomohara on it?
Yes, yes. This is Tomohara, first paper and second paper. Yes, yeah. Yes.
Okay. The next question is from Amanullah, who says, have you looked into the relationship between L-type channel distribution and arrhythmias?
What I can say, I can say this, yes, because we use this channels work and we use this channels just results, and Natalia simulates this and try to do the just simulate this on the whole heart. In this situation, yes. Because the problem, because the idea now, we more and more start to work with the heart failure patients and the different patients. And what we are trying to track, how often these patients have arrhythmia and what the arrhythmic mechanisms and what the different arrhythmia they have during the heart failure. And this is our future work. We just really have a big cohort of patients, and we try to have this link and study more closely how our small nanodomains can affect to the different type of arrhythmias. Yes.
Okay.
Can I
yes,
ask a follow-up question? Yeah. Hi, this is Aman from George Mason University. We looked into the channel activities from RYR and L-type in our modeling. And, we can see the sparks are a little longer and, bigger, the duration is different, everything is different. But it's when we looked into the detail, we never seen any effect on the on the arrhythmia, like APs. APs pretty much like a little bit, change the shape, a little bit change, but there isn't like, alternate or EADs or DADs kind of pattern. We don't see that in our modeling. So can you please comment on that?
I don't know, because I cannot really answer about the modeling work, because it's just mostly Natalia work, and I can just be I can just comment on my practical work.
What do you think about experiments? Do you think?
Because I think it's about experiments, because what we are doing, we are just trying to check and because honestly, I don't have enough time to explain what we are doing. We are just really have a different topography of the cells with the normal T-tubules and crest, and abnormal with the heart failure. What we are doing, we use our pipette and we measure, we pick up the calcium channels from the direction from our area of interest. And our area of interest, this is a T-tubules and crest. So do you see my just could you see my presentation?
Yes.
Okay, let's say just show fast what we are doing, because we okay, so we have here the cell, and we have here the cell with topographical image, and we try to patch with this already clipped pipette. When we can cover the T-tubules, we try to patch in the two different directions, in the just crest and in the T-tubules. and we measure and we record the functional channels. And when we can just potentially, when we see the high open probability of these channels, we try to block these channels with the different blockers. And potentially this is open probability of our channels. And during the infarction and then failing heart, we have a high open probability channels. And we try, in order to check the phosphorylation of the channels, we try the different blockers. In the situation in the different patients' cells, our blockers are different. We can block PK, we can block CAM kinase. And in the different diseases, we have a different blockers. And after that, the simulation, this how they do the simulation, they try to check first of all, they try to use this characteristic of these channels, open probability channels, and after that, it's you can just check the paper, especially in circulation research, the supplementary. There's a lot of additional steps how they just go to this simulated saline and why they have this reentry and why they just got this using our data in the high open probability L-type calcium channels. Yeah.
I see. Thank you very much. Thank you.
Okay, I think this is the last question. So thank you again, Yulia, very much for a fantastic talk, and also to everyone, a very wonderful holiday season. Enjoy the times and stay away from Corona. Okay. Bye.
Thank you very much.
Yulia. Thank you all.
Thank you. Thank you.
Bye.
Bye-bye.
Thank you.
Oh, Yoram, do I need to stay or I can go?
I don't
I
know.
think we're okay.
No,
Okay.
you can log off. Yeah, it's
Yeah,
time.
thank you.